everybody, and good morning from India. Welcome to another episode of The Living Algorithm. So, hi Sushant, good morning.
Very good morning.
So, folks, what if cancer isn't just a genetic disease but also an electrical one? Today, we are diving into the bioelectrome, the electrical conversations happening inside our cells, and how mitochondria, the powerhouse of the cell, might be right at the center of it. So, Sushant, my first question to you is, how do the electrical properties of cancer cells differ from healthy ones, and where does mitochondria fit into that picture?
Okay, we have to understand from the beginning, let us have a very clear understanding of before I answer your questions of the role of bioelectric patterns and bioelectric memories, right?
Hmm, hmm,
So we are born with one cell, and then your cells divide, and we have then liver cells and heart cells and brain cells, you know,
hmm,
uh, gut cells and a lot of things, right? And they do not know, these cells do not know which organ or tissue they belong to, right? So every cell, you know, operates in a very, predefined, area or predefined, computational capacity. So what I mean by computational capacity is that it's like an operating system, right? So essentially, like a computer, like a laptop, you receive an input, you process it, you actually analyze it, interpret, and give an output signal, right? So every cell
true.
has that sort of, in architecture where it is receiving environmental cues, it is sensing it and giving some output signal and giving some response, right? So that's a very limited capacity, right, of every cell, and that was when there was unicellularity before humans were born, 2 billion years ago, 2 billion years ago, right? So every cell was a single cell organism, and they were just, pursuing their own selfish goals, right? Uh,
Hmm, hmm,
and then, a larger cell, a larger organism engulfed a smaller one, and instead of using it as a source of fuel, it maintained a symbiotic relationship, and this smaller organism is with mitochondria, right? So essentially, that's how we became a multicellular organism, right? Humans, mammals are multicellular organisms. So coming back to the thing that, every cell inside our body have this sort of computation capacity. So for instance, your pancreatic beta cells may have an, computation capacity of sensing and sensing the environment and releasing
hmm,
insulin. So
hmm,
so again, that is just an example, right? But what happens is that these all cells are bound together by a network, right? So there's a bioelectric network, there's a bioelectric electric pattern, so they are connected together. And when this whole cell are connected together, there is some called a collective intelligence, or there's an inherent intelligence, and now this collective, or this entire family of 37 trillion cells is now this collective knows exactly what is going on.
hmm,
And then and then the cognitive capacity or the computational capability of the entire network is larger than that of single cell, right? And they cooperate with each other, right? So they always take it as a give and take relationship, right? So essentially, they are not defecting, they are working together in return of the metabolic learning and effort which other cells have done, right? So they're getting the learning from the other cells, right? And the cognitive capacity expands, right? So rather than just pursuing a local goal, they all work together towards towards our anatomical goals, towards keeping our body healthy, and uh, sort of maintaining that normal structure. So single cell do not know where your finger, head, or ear is, but collective does know, right? So essentially, that's how they pursue anatomical goals, a goal-directed behavior to keep you healthy, right? And again, how they communicate is through bioelectric memory. So they have bioelectric memory, through which they communicate. Then they could read the the nuclear DNA, the genome, that's again standard operating procedure, SOP, or instructions for how the body should function, right? So the cells are basically agents, and they read those genetic instructions. They read the bioelectric patterns. They listen to the signals sent by other cells, and then they take decisions. And when there are mutations or inborn errors in metabolism, as well as there are certain genetic defects, cells, when they communicate, they could even override those instructions or bypass those instructions, right? And take the right decisions for our biology, right? So till the time these cells are working together, they have a bioelectric memory, they are communicating together. And now coming on to your question, right? So every cell is connected via protein called gap junctions, right? So they are connection proteins, and these, so every two cells, take it as a yatch or something like that, right? So essentially, every two cells are connected with a gap junction, right? So essentially, they communicate, and how they communicate, they send out the ion channels, right? So they release, we know there are ion channels, there are calcium channels, there are chloride channels, there are potassium, sodium, right? Yeah, so they release these, which goes to gap junction. They communicate. So if a cell is under stress, right, they communicate with other cells, and then the cells to take the right appropriate responses. So essentially, there is an information coming, there is an information being received, being processed, right, being transduced, and decoded by other cells, and then converted into certain responses, right? And that makes a shift to your allostatic state. So essentially, if you have, probably if you have hyperglycemia, it is a movement to a new state, because your cells, your body try to keep you in an best possible way in a homeostatic state, right? So essentially, that is something which is important in terms of understanding the bioelectric patterns.
That's pretty interesting because you know what? Michael Levin and others talk about the bioelectrome as a kind of a software layer that tells cells what to become, right? So let's say if that software gets corrupted, so does mitochondria become both victims and amplifiers of that problem?
So what happens is that there are few things we have to remember, that first thing is, we all consider mitochondria as a powerhouse, which it is, right? It
Hmm.
is. It receives the electron goes to electron transport chain, all complex, and it is at the end of the last complex 5, it jumps onto oxygen, and ATP is produced. Right? And we
Hmm.
inhale oxygen and leave carbon dioxide. So the byproducts of this ATPs are obviously water and carbon dioxide. So essentially, so that is one function of mitochondria. But
Hmm.
mitochondria also controls your cells. So it decides whether cell has to divide, cell has to stay quiescent, cell has to grow, or cell has to die, right? So mitochondria controls the apoptotic signaling of the cells, right? So whether a cell has to grow indefinitely, or to some extent maybe keeping quiet, or dividing into different more cells, right? It all is driven by instructions given by your mitochondria, right? And mitochondria also controls your calcium circuits, your ion channels, right? So basically, the movement towards the movement of the calcium ion in and out of the cell is actually controlled by your mitochondria, right? So you have to understand that our cells from inside is more negative than from outside, right? So your calcium are positive ions, your sodium is positive, your potassium positive, chloride is negative, right? So essentially, we need potassium inside the cells, right? So essentially, there's a sodium potassium pump, which actually works when there is enough ATP being transformed by your mitochondria. And this pump makes sure that there is more, for every three sodium ions coming out, there are two potassium ion going in. So obviously, it maintains that less positive charge inside the cell. And then again, calcium remains more outside the cell, right? Because calcium is highly positive. Now, calcium ion is very important, because when the electrons are transferred from NAD converts to NADH or FADH2, probably depending on whether it's carbohydrates or fatty acids. So essentially, uh, calcium gets inside the cell, and calcium is like a fuse, like a charge, right? So it activates enzymatic functions that helps in transformation of these electrons into energy, and then calcium goes out of the cell of the mitochondria, right? So basically what happens is when we discussed about the energy resistance in the last talk, right? So essentially, the external stressors or certain resistances to energy transformation, right? Uh, certain resistances are important because it helps you transform the energy. But if there's too much of resistance, it actually impedes the flow of electrons within the electron transport chain, right? And it's like, understand, there's a highway, there's a lot of traffic, right? So essentially, some traffic moves back, electrons start moving back, right? And they jump on the molecular oxygen, which leads to production of reactive oxygen species, ROS. And then there's ROS, and there's no electrons to be transformed. There's calcium inside the cell. So again, it leads to calcium overload, right? And when it is a chronic calcium overload, right, it could act as an electrostatic barrier and could block the gap junctions, right? So gap junctions are the way the cells communicate, right? So when it blocks the gap junction, cells cannot communicate, and that communication is broken, right? So this is independent of your DNA. And in fact, what we have found is now that your mitochondria controls or instructs to turn on and turn off nuclear DNA, right, which is a human DNA. So it tells your nucleus which genes have to turn on and turn off. And this is done basically in around 70% of your genes. Right? 70% of your human genes, around 15,000 human genes, are receive instructions from your mitochondria. So again, mutations are a different concept. Probably I can discuss a bit later on that. But let us first consume this. What probably it's it is
I
important to
think
understand.
this is a very different perspective on how generally they assume that cancer, the reasons behind the cancer, right? Now, my next question to you is that there's a growing interest in targeting mitochondrial function and membrane voltage as cancer therapies, right? So, how close are we to turning bioelectric or mitochondrial resets into real treatments? And what is your take on that?
So you're talking of the Indian market or the
Let's
American
say
market?
global. I mean, because I am pretty sure that in India these therapies don't even exist. So let's talk global. I mean, because of course something which is invented in the US it will eventually come to India years
So
later. But it will.
there are people in US, Canada, Europe. There are people getting to research on this, like you mentioned, what Michael Levin is doing research on the bioelectrome on building regenerative
Hmm.
drugs that could probably so before that, understand that when there is a breakdown and close of gap junction and the cells detach from the cell collective, and then for that cell they have to grow, right? And they fall
Hmm.
back on the ancient pathway, which is growth and proliferation, unlimited growth and proliferation. And they want to grow not in next 2 minutes, but next in 2 milliseconds. So then
Hmm.
comes to biochemical reaction, they'll upregulate. And again, here the role is when there's this thing happens, mitochondria uh, through retrograde signaling tell your nuclear DNA to overexpress oncogenes that could bring all the fermentable fuels inside, which can be used to generate energy for that rogue cancer cell. Right? So essentially,
Hmm.
when the cells are detached from the cell collective, they start pursuing their own goals, their own limited cognitive goals, and for other cells are cognitive the other cells are external environment, and this is what cancer is, right?
Hmm.
So essentially, the point is that these researchers and scientists are basically working on how do we can make these cells, instead of killing them, which has a lot of collateral damage, which is standard of care practice, that you take up therapies and drugs and chemotherapies and a lot of these toxic therapies to actually try to kill those mutated genes and those tumors, right?
Hmm.
Unknowing that there is a huge intertumoral heterogeneity, right? So same tumor may have different genetic mutations, again, you cannot target everyone because you may die of that toxic therapy with these drugs. So basically, there are a lot of these, mutations which can bypass, right, and start growing, right? So essentially, that is one of the gap in the current standard of care therapy. So the people are thinking from a different perspective. How though rather than killing cancer cells, why don't we make them rejoin the cell collective, right? This is something very fascinating because these apply to both neural cells and non-neural cells. So basically, we think that the intelligence only lies in the brain. That is what we think, right? But cells have their own intelligence, these bioelectric pattern memories which they store, right? They can refer to them to take decisions. So essentially, there is an inherent intelligence not in the neural, not only in the neural network, even before the brain cells were formed. So they are the scientists and researchers are exploring this, this concept of identifying how they can use the innate intelligence of the cells of the collective to make them rejoin. So again, these would be, I think, in globally these should be commercialized in next 4 5 years or maybe much earlier. Right now, a recent report, recent paper it came out. And interesting to share is that a recent paper came, which I also shared on LinkedIn, that the cells are releasing low intensity light emissions, right?
Yeah,
Okay.
right.
So
Yeah,
again,
we
the
spoke about that
right,
the other
right. So
day.
essentially, they
Yes.
are what it is. So basically, they are diffused when they produce ATP and then they release ROS as a part of normal aerobic metabolism process, right? And again, these actually are also a way of communicating. So even during the stress responses, when the cell is experiencing inflammation or injury or infection, they also release light signals. So again, again these also there are different varying patterns, right? So these different patterns are different in healthy and cancerous cells. So to answer your question, that this is one of the way you can look at, and even other biochemical differentiation is obviously cancer cells ferment, right? They ferment glucose and glutamine. They don't respire. So again,
Yes.
this is more inefficient way of transforming energy, right? So they produce very less ATP in fermentation vis-a-vis respiration, what we call is oxidized phosphorylation, right? So essentially, there is again a differentiating factor, and again for that they overexpress a lot of glucose transporters and to ferment these. So they cannot ferment fatty acids. Right? Fatty acids are stored in lipid droplets. They cannot use it again, and that is done even in the presence of oxygen because mitochondria is corrupted to perform that transformation process. So again, this biophoton thing or the low emission light emissions can help differentiate between an cancer and a healthy cell, right? Uh, right. So again, that is another way that could replace maybe invasive biopsy that has an inherent risk of converting benign cancer to a malignant tumor cell, right? So essentially, that could be another important invention which could really help in terms of
Yeah,
identifying cancer.
that's true. I mean, that will be the best thing humanity might end up discovering or inventing because this will be non-invasive and it won't require a lot of lab work, right? Just like you go for an MRI, it will be similar to that, right?
But actually, it has to be
So,
proven because otherwise, still the current prediction model space see that if you are detecting cancer, probably you have to get for a PET scan. If you are detecting for a colon cancer, you go for a colonoscopy and all those things. That would reconfirm for healthcare practitioners. It would reconfirm because that's a standard of care right now. But obviously, this cannot work in isolation. This has to be combined with standard of care. So you can't wait for colonoscopy to show certain aberrant signals, right? But you can identify the early signs, and then you can revalidate it, and then you can adopt certain actionable interventions that could prevent the cancers from growing.
I have a very interesting question for you now. Let's say if we could listen to the electrical chatter between mitochondria and rest of the cells in real time. So, just a thought. What do you think we would hear in a tumor versus a healthy tissue?
So, mitochondria of a cancer cell would be shouting that, I'm I'm being attacked. I'm not working well. My legs are not working
Mm.
well. I can't listen. No,
Mm.
probably I can't. I cannot. I have to give me more glucose and glutamine. I need to use that. So, and essentially, that is one of the way. Cancer cells, mitochondria. Because the point is, we are again focused on the killing those mitochondria, right? Because they have lost the ability to do their main job, which is again transformation of energy. But they can't send signals. They tell nucleus to start oncogenes. So a lot of people who are looking at oncogenes
Mm.
overexpression is because of mitochondria telling nucleus that I can't produce, I can't do use oxidized phosphorylation to generate energy, right? Start fermenting, right? So
Mm.
that's a
Mm.
kind of
Mm.
message.
Mm.
So these are generally retrograde signaling and cytokines and a lot of these
So,
things.
what kind of frequency would tumor cells have? I mean, what
I
is
can't
it?
answer that. I have no
Is
answer to
it
that.
okay? Right. So, I think people are still working on discovering those signals, the bioelectric signals, right?
Because these lights cannot be seen from naked eye. If you talk of biophotons, this cannot be seen from naked eye. There are
I
specific
know
instruments that can.
they're very minute. I think you would need some uh, you will need very fine technologies to understand such signals, right? So I totally understand that. So my next question to you is like a closing reflection on this discussion. So what is the single most surprising or hopeful idea that emerges when we start thinking about cancer as a both a metabolic and an electrical disease?
We're talking of how to resolve it.
Yeah, of course. I mean, given the current times that we are
So
in and the
so
forward-looking inventions, so
mhm.
what would you think be
so
the
so which
ideal idea?
what is possible right now is, again, I'll say quasi possible, because I think not many people are focusing on understanding the energy inputs of the mitochondria, right? So essentially,
Mm.
the focus should be to first understand, what mitochondria is doing, how it is interacting with various external cues, whether it is your, environmental toxins or people you hang out with, or even the food you consume, or even the microbiome mediated, metabolites, right? How those
Mm.
is impacting the ability of mitochondria to transform energy, right? Because the higher these resistances are there, right, the higher energy resistance, and lower the ATP production, higher the ROS inside the mitochondria. And then again,
Mm.
this makes mitochondria release cytokines, which communicate with other mitochondria. And again, there's a reallocation of energy, because there's a limited energy budget what we have, right? So essentially, we reallocate the energy to respond to external stresses rather than growing and maintaining and repairing cells and body, right? So essentially, when this is
Mm.
then there's too much of systemic inflammation and all these infections and insults, right? There's a state of hypermetabolism where we spend a lot of high energy, and then that's how can your mitochondria resort to your cells resort to fermentation model, because there is they cannot, there's a very high energy expenditure, right? And they
Mm.
have to
Mm.
resort to other models to in mitochondria again become dysfunctional. So again, they have to, when there is no energy to, then the mitochondria will resort to the fermentation model, which is using lactate and succinate and all those things, right?
Mm.
First thing is obviously focus on the mitochondria, the energy piece of the mitochondria, right?
Mm.
Directly, indirectly, you should focus on measuring it, right? Second thing is obviously the bigger dreamy picture would be obviously not killing cancer cells and making them rejoin their family of other cells. And again, you
Mm.
have to the
Mm.
ways you have to maybe resort to certain drugs or certain molecules that could
Mm.
open up the ion channels, right? Reinstate the maybe rebalance the ion channel equilibrium, right? Or open up gap
Mm.
junctions that can initiate the reconversation between the cells. Right. So again, that could be another major, treatment protocol, right? Apart from the standard of
Mm.
care. So that If you can identify those things, probably standard of care may be required at a very minuscule level, right?
That's true. I think this is a very progressive approach if taken by the mainstream healthcare industry, right? So, Sushant, thank you for such a deep and very interesting discussion. And any last words would you like to add here?
See, a few things you have to understand that type 2 diabetes is a precursor for cancer, and type 2 diabetes also, so every single disease is
Hmm.
caused by corrupt mitochondria being corrupted, mitochondria not
Right.
able to perform their functions, right?
Hmm.
So even in insulin resistance, which we say is causing diabetes, again, insulin resistance is a symptom. But insulin resistance also happens because of high energy resistance within the mitochondria. So it's an anti-defense mechanism of mitochondria to prevent from expressing certain root 4 receptors, right? So they're saying that, I don't need that much glucose. I'm overwhelmed with a lot of this fuel. So please do not come here, right? So again, so this is that's how you have your glucose cannot get inside the cell, and that's how you have hyperglycemia and insulin resistance going forward, right? So again, type 2 diabetes is a precursor for cancer. This is very simple explanation that the common cause is the breakdown in the mitochondrial energetic processes, right? So mitochondria,
Hmm.
it's a cell danger response, right? So you can take insulin resistance as cell danger response, right? And that's what, so over a period of time when there's a hypermetabolic state, and when the mitochondria is not able to transmit these ion channel circuits, right? There's ROS, this leads to calcium overload, this leads to disconnection of the bioelectric network and distribution network of the cells, and then we have cancer, right? So you know uh, this is something people should focus for type 2 diabetes. Now, do not you know, measure your glucose, measure your insulin levels first, right? Look at your oral macrobiome, gut macrobiome. Your oral is the starting point of every single disease. Oral health is the starting point of every single disease. Fix your oral macrobiome, fix your oral health, fix your gut health, right? Understand your mitochondria, right? Uh, uh, have good amount of healthy fats, right, which is required for maintaining the cellular integrity, right? So
Hmm.
I'm going to do a short on this, that uh, why we are too much obsessed with high protein, why not high fats, essential
Hmm.
fatty acids, right? Which
Right.
we do not adopt, right? So
Hmm.
that is something we have to think, right? So these are the few words probably which is important to understand, because if you are type 2 diabetic, or if you have hyperglycemic, you have an insulin high insulin levels, right? You have erectile dysfunction, sexual dysfunction, right? Or you have high blood pressure. All these are early signs for different forms of cancer.
Hmm. So thank you, Sushant, for your time and sharing with us the research, everything that you have shared with us based on your research. And folks, thank you for listening to our
I
last
just want to
episode.
add here, right? So these all are insights from the research done by Professor Michael Levin, Professor Martin Picard, Niresha, and
Hmm.
so
Hmm.
so uh, Professor Nirosha. So essentially, all these are published somewhere. So this is all something we have referred there, and we are sharing with you, right? Based on our understanding of how it works, right? And we are also trying to implement this in our cancer model. So
Hmm.
this is what Bioelectrome was. Next week, Sakshi, we should discuss about fermentation.
Yes, of course. Our next episode will be about fermentation, fuels, and how cells ferment, how cancer cells start fermenting, and they become cancerous, right? So thank you, Sushant. We will be back with another episode soon.
Thank
Thank you.
you. Bye bye.